SelectSpy Research develops numerous kits for fast and effortless targeted gene integration into different cell lines. This protocol demonstrates how one single transgene expression cassette is integrated into one unique transcriptionally active site within the genome. The first step of the procedure consists in cloning your gene of interest into an integration matrix by standard cloning methods. We have many types of integration matrices to choose from to control the level of expression, as well as to tag your gene of interest. To learn more, please look at our Plasmid Integration Matrix store. The integration matrix contains flanking regions of homology necessary to drive homologous integration and targeted integration at one precise and unique genomic site. The precise map of the integration matrix in the related datasheet helps you to choose the most suitable restriction enzyme for your cloning. Successful targeted integrations have been achieved with 8.5 kb sequences with no loss of efficiencies. We also have integration matrices that allow two genes of interest to be cloned into the same plasmid and subsequently integrated into the same locus. As soon as your integration matrices are ready, you can prepare your cells for the co-transfection together with a meganuclease plasmid supplied with the kit. Before proceeding with the co-transfection, we advise you to verify that your cells are in good physiological conditions and at appropriate confluencies of between 60 to 70%. If these conditions are met, you can proceed. Co-transfection is to be carried out at room temperature in a sterile environment in a laminar flow hood in order to avoid any contamination. 1 day prior to transfection, 200,000 cGPS CHO-K1 cells are seeded in 10 centimeter tissue culture dishes in complete medium without hygromycin. After a 24-hour incubation period, the cells are ready for transfection and the transfection mix can be prepared. To obtain optimized results, we strongly recommend you to thoroughly follow the described protocol supplied with the kit. In parallel, do not forget to do the positive control experiments with the provided control matrix. Hours after transfection, replace medium with 10 mL of complete medium supplemented with G418. At day 3 and then at day 6, replace medium with fresh complete medium supplemented with G418. During these days, you will be able to detect a high percentage of dying cells, which indicates that the selection process is working. At day 8 and day 10, replace medium with fresh complete medium supplemented with G418 and puromycin. More cells will die during this second selection step that ensures a correct and balanced targeted integration of the transgene at the desired genomic site. Once you doubly selected the clones, they have to be individually picked and transferred into a 96-well plate. If you do not have access to a picking robot, you can easily do it by hand. Around 13 days after transfection, single colony clones with sufficient size are picked and seeded in a 96-well plate containing fresh complete medium supplemented with G418 and puromycin. Regularly, check the cell confluency of each individual 96 well. When confluency is reached at around day 17, transfer trypsinized cells in a 6-well plate in fresh complete medium supplemented with both selection agents. About 4 extra days are needed to amplify each single cell clone in a 6-well plate format. An additional 3 days are needed to reach confluency after transfer to 10 centimeter tissue dishes. The number of days is indicative. It is recommended to check cell confluency at each step. A targeted integration event occurs in up to 98% of the clones. A simple PCR test will confirm targeted integration in most of the selected clones. A correct targeted integration of the gene of interest can be identified at the molecular level by Southern blot. By PCR, you will be able to detect targeted integration events only, but by Southern, you will also confirm that the targeted integration events are in a single, unique copy. The matrix restriction map in the related datasheet will help you to choose the adequate restriction enzyme for your gene. If the radioactive segment corresponds to the size of the junction, the targeted integration of the gene of interest has been successful. This protocol has been extensively applied by our product development team and gives optimal results. Thus, we strongly recommend to thoroughly follow the described protocol supplied with the kit, even though it could slightly differ from your own practices. Do not hesitate to visit our PIM store or to contact us for assistance.